All right, well, I'd like to welcome everyone to today's edition of the Polyploidy Webinar. This is our last webinar of the semester and of calendar year 2020. Today we have a PhD student with us, Veronika Konečná. I had it earlier and sorry, Veronika. Veronika is a fourth-year PhD student in the group of plant ecological genomics with Filip Kolar at Charles University in Prague in the Czech Republic. During her PhD, she's had a number of great collaborations and research experiences in other folks' labs, including those of Dr. Levi Yant and Professor Christian Perisseau. She is generally interested in the drivers and evolutionary consequences of whole genome duplication and parallel evolution in extreme environments such as alpine and serpentine soils. And for this Polyploidy webinar today, she's going to present the main project of her PhD research entitled the repeated recruitment. Repeated recruitment is the dominant source of adaptive variation in autopolyploid Arabidopsis Aranosa. And with that, I'm going to let Veronika take it away, and I'm really excited to hear about this cool research.
Okay, Mike, thank you for the introduction and also for the organizing such a fantastic series. So today, I would like to talk about autopolyploid Arabidopsis Aranosa and its adaptive variation sources. And this story will appear soon on bioRxiv. So let's start. Long-standing controversy surrounds the potential impact of polyploidy on adaptation. Besides immediate phenotypic and genomic changes following whole genome duplication, and also a great colonization potential of polyploids, as was shown in these studies, it's still unclear how adaptation proceeds in autopolyploids. So on the one hand, we know that polyploids are better in masking of deleterious alleles. They harbor large genetic diversity and number of mutational targets is multiplied. But on the other hand, the changes in allele frequencies are slower, which may retard the adaptation. And recent progress in theory and simulation propose a solution to this controversy, that polyploidy may promote adaptation under rapid environmental change. And the rapid environmental change is mainly powered by the availability of genetic variation, which is the strong advantage, obviously, of polyploids. So what I mean by a rapid environmental change? So I mean, for example, colonization of challenging environments such as railways, alpine environment, or toxic edaphic substrate such as mine tailings or serpentines. So it's still largely unknown what is the genomic basis of such a polyploid adaptability, and whether the adaptive alleles are primarily sourced from the standing genetic variation or de novo mutations. So I would like to address these questions in this talk. Okay, so let me briefly introduce our model environment, which is Serpentine. So it's one of the greatest challenges for plant life. It's highly selective environment for which is typical elevated levels of toxic metals such as nickel, cobalt, chromium, then also high concentration of magnesium and reduced calcium to magnesium ratio. So calcium is not easily available for plants. And what is further important, that serpentines occur in the landscape as a scattered islands, which promote parallel adaptation, and they served as a natural replicates in natural laboratory. So if plants want to survive there, they have to adapt. And there are several possible sources of adaptive variation, including selection on de novo mutations, or standing genetic variation, or introgression. And here I interpreted those last two as shared origin, because both processes operates on the single mutational origin. So now let's introduce our model plant system, which is Arabidopsis Aranosa. It's a well-characterized diploid autotetraploid species with large outcrossing populations with high genetic variation. And tetraploids are widespread across Europe, and they also colonized serpentines compared to the diploids. And as a proof of the concept, polygenic multitrait basis was shown by Arnold and colleagues only in one tetraploid serpentine population. So it left unknown whether the genes are reused by parallel selection and what are the sources of adaptive alleles. And as we know about other tetraploid serpentine population, it's ideal model system to study. So now let's start with the first chapter about parallel serpentine adaptation. So we firstly ask if serpentines were colonized independently in each geographic region. So we sampled 5 serpentine and geographical proximal non-serpentine populations, all of them tetraploids and all of them from the Central Europe. We used the whole genome resequencing data, and here on the left, as you can see from the allele frequency covariance graph, you can see that population clustered based on the geography, not based on the soil type. And the same result was confirmed by coalescent simulation in FastSimCol, where we tested parallel versus single origin scenario. And for all pairwise comparisons of serpentine, non-serpentine pairs, the best, the scenario with the highest support was parallel origin. So to sum it up, that we are dealing with 5 parallel cases of serpentine colonization by tetraploids in Central Europe. So then we were really interested if serpentine plants are really adapted. So we asked if the same environmental trigger can cause similar adaptive responses. So to answer this question, we conducted a reciprocal transplant experiment in common garden condition. And this time we used 3 population pairs and soil from 6 natural localities. Here you can see the results from the first part of the experiment when the plants reach the maximum rosette sizes. And here from the graph on the right, you can see consistent disadvantage of non-serpentine plants in wild, where they were cultivated in serpentine soils. And the same is also obvious from the illustrative photos, where the dots denote the origin of the plants. Okay, so now let's continue with the second chapter about parallel genomic footprints of selection on serpentine. So firstly, I would like to show you our methodological overview for the detection of directional selection to infer genes which are under selection in serpentines. So we basically use 2 complementary approaches. Firstly, environmental association analysis, where we were searching for significant associations between allele frequencies and individual concentrations of particle elements in the soil. The second approach, window-based FST scans. So we detected 1% of FST outlier genes. And here we are back with 5 population pairs. So we ended up with 5 lists of FST outliers, kind of replicates. So those were really permissive lists. And to further refine these candidates, we took only these which appear at least in 2 population pairs and call them as a parallel FST ones. We also checked the distribution across the genome of these candidates to see if they are not limited, for example, to the regions with the low recombination rate. And then we made a final refinement by overlapping these candidates from these 2 approaches. Okay, so we ask how frequently does evolution repeat itself at genomic level. So we detected a substantial parallelism. And as I told you in the previous slide, we ended up with candidates from 2 complementary approaches. We overlapped them, and we found 61 serpentine adaptation candidate genes. And here in the FST scans for pair 4, you can see the distribution of these candidates across the genome, and you can see that the basis of serpentine adaptation is polygenic. And what is also important, that we found some relevant biological processes and molecular functions enriched. So, for example, transferase activity, chemical homeostasis, or cation transport. Okay, so now let's go to the final chapter about sources of adaptive variation. So we detected substantial parallelism, and we used that parallelism to test for the sources of adaptive variation. So to infer the sources of variants for our serpentine candidate genes, we used the new model-based approach, DMC, by Lee and Coop, which was specifically designed for closely related populations. And we found that the majority of the variants of our parallel candidate genes has shared origin, with the only one exception of 2 portion loci, calcium central channel. And I would like to tell you more about this interesting candidates. So we looked further in a sequence variation in this parallel candidate loci, and we found 2 de novo alleles, tyrosine and leucine, only in serpentine populations in this pre-alpine region. And here from the element, you can see that those non-synonymous changes are in high frequency, and they are differentiating those serpentine populations from the other ones. Then, to confirm that those are really private to the serpentine populations, we checked across all available populations of Arabidopsis aranosa from the World Range, and also sister species population of sister species Arabidopsis lyrata and haleni, and we confirmed that those are really private for serpentines. So we expect some functional impact of these alleles, and our colleague Sean Bray added the structure homology model, and here you can see a residue in red which sits directly in the main pore opening of this channel, calcium channel. So we can expect that it has a functional impact, which means it has impact on the selectivity of calcium through this channel, which is really relevant for the serpentine adaptation. So it brings me to the end, so let's conclude. So I showed you repeated parallel adaptation of Arabidopsis aranosa to serpentines. We detected substantial parallelism in serpentine candidate genes, and we found that repeated selection from the shared pool of alleles is dominant mechanism, but also rapid sweep of de novo mutation is possible. And as a take-home message, polyploids can very rapidly adapt to environmental extremes, calling on both existing variation and novel polymorphisms. So I would like to thank you to all of my collaborators. Actually, it was a nice collaboration across so many institutes and universities. And mainly, I would like to thank to my supervisor, Philip Collage, and to my main collaborator, Levi Yant. And you for your attention.
All right. Well, thank you, Veronika, for a wonderful talk. We'll take some questions now. As usual, we'll put those in the chat box, and folks can turn on their audio and video and ask those as well. That often makes it a little bit more enjoyable. All right. Looks like, well, that's Justin saying great talk. All right.
Thanks.
Alan asks soil analysis.
Yeah, of course, we did the soil analysis using inductive couple mass plasma spectrometry. So we were checking for the concentration around 20 elements, and then we detected the elements which were differentiating serpentine and non-serpentine soils, and it was calcium, magnesium, nickel, cobalt, and that's all. So I use concentration of these elements then in the environmental association analysis. And we have these data available for each individual which were resequenced. Mm-hmm.
All right, I think that answered the question. Justin Conover is up next.
Hey, Veronika, great talk. I really enjoyed it.
Yeah, thanks.
Do you think that so you found that most of the alleles that are responsible for this serpentine adaptation is shared, so it's not independent events. Do you think that's because these populations just aren't differentiated enough, and so there's just a lot of shared variation? Or do you think it's something to do with the fact that you had to use FST, so you're really only going to be looking at sites that are shared between different populations?
So I would like to tell you more about the parallel selection. They has the shared origin, but it doesn't mean that they the alleles were not parallel selected. So here in blue, it's kind of a selection. It's kind of an environment under selection. And you can see that independently in those two populations, the variance, the alleles increase in their frequency, like parallelly. So it's still a case of parallel evolution. Yeah. And regarding the differentiation, so I think that I didn't mention that the mean divergence time for serpentine and non-serpentine populations from the differentiation was around 7,000 of years. So it's really like post-glacial. And yeah, there's a low divergence, but still they are pretty differentiated.
Okay, great. Thank you.
Yeah, you're welcome.
All right, next question is from Fernando. If you want to turn on your mic and ask. Yep. Or we just read it here. So Fernando asked how you tested for integration. Yeah, what approach did you use?
Yeah, okay.
No problem.
So what kind of introgression do you mean? So because in the DMC approach, by migration, I mean the migration between adapted serpentine populations, right? Okay, so that was migration, yeah. So migration between the serpentine populations. And then, of course, that there is possible introgression from another species, Lerata, but now I'm talking more about ancient, older introgression, because our populations are not were not sampled from the hybrid zones. So I think that we don't have to worry about introgression from Lerata here.
All right, I had a really quick question. Well, I think that answered that one. So how common, do you have a sense of how common the alleles associated with that you find associated with the serpentine plants, how common are those in the non-serpentine populations? So do you have an estimate of the frequency of those alleles in the non-serpentine populations? So I'm just thinking about how much standing variation, how common is that standing variation that was used during adaptation, or you think was used during adaptation to the serpentine soils?
Yeah. So it's an interesting question. It depends on the geographic region. So as I observe from my data, there are regions where this standing genetic variation responsible for the adaptation, it's not really easily available, because you can see Arabidopsis Aranosa plants just around the Serpentine site, but they not colonize it. Then the situation is a little bit different in Austria, in pre-alpine region. But still, as I showed you from the results from the transplant experiment, you can see that those non-serpentine plants were not able to have a nice big rosettes. They were almost dying. They were surviving, but really with the tiny rosettes, so they were not very happy.
So it must be pretty rare then, I guess. That's even though it's probably standing, it's pretty low in some of these cases. I was just curious, thinking about the selection gradient for the sweeping those alleles.
Yeah. Actually, it's interesting that serpentines are really like islands. There is no continuum gradient, so the boundaries are really sharp.
Yeah, cool. All right. Well, are there any other questions out there for Veronika this morning?
It's not even morning for all of us.
Or I apologize. Yes, I think it's only morning for a few of us still.
I just wanted to ask, what do you think that keeps the variation, or the standing variation of those serpentine beneficial alleles low in the non-serpentine population? Because from your common garden experiment, it seemed like there is not much deleterious effect for having this allele. So it seems that it's universally positive. So what's going on here?
Yeah, that's true that I can go back. So there was no any trade-off regarding this vegetative fitness indicators for serpentine plants, and they were cultivated in non-serpentine soils. So yeah, maybe still, because we should keep it in mind that the divergence time are really low, so maybe it's behind it, and some kind of a balancing selection may play a role.
Yeah, that's a great question, Camille. I had the same idea. That's partly also what I was thinking is how much. Why don't we see it flowing backwards too, right? From the serpentine to the non-serpentine adjacent populations, because it doesn't seem to have a huge cost, at least in this analysis, but it may still have some fitness cost that isn't measured here as well, right? They perform okay, but I'm sort of curious if
but
there's some other dimension, right? Where yeah.
Yeah, that we should take in mind that in this experiment, we did not account for example, competition. So maybe if we cultivate serpentine plants in non-serpentine conditions with the competitors, the results can be different. Yeah. So there is other question. What do you think the contribution of polyploidy here? I guess both N and S are polyploids. Yeah, so all of my populations are autotetraploids. And I'm not sure if you noticed that I told you that only autotetraploids colonize the serpentines. So far we don't know about the diploids of arenosa on serpentines, although in Balkan Peninsula, in Balkan regions, there are huge serpentine barrens. And diploids occur there, but so far we don't know, or we don't have any evidence that they are explicitly there in serpentines.
This is Paul Allen. If I may interject, the reason I was invited to this talk, and this is the very first polyploid talk that I've joined, so thank you very much, and please excuse me if this is too basic a question, but we have a discussion group of amateur biologists every week, and this week we're talking about polyploidy and versus diploidy. And I can easily see how polyploidy is advantageous to adapting to new or rapidly changing environments, as was demonstrated in this research. Thank you very much. But when we look out on the wild world about us, it's clear that diploidy is the dominant feature we see. And it occurs to me that although we've identified many advantages to having polyploidy in terms of abundant genetic material to work with and to create novel metabolic pathways and the like, there must be a cost to maintaining polyploidy, as it would seem that most of the plant species in the world have some ancestral fingerprint of polyploidy in their evolutionary past, and yet they have regressed to being a diploid dominant feature in most plant genera. I wonder if someone could speak to that. Why is diploidy so dominant in the landscape when there appear to be robust advantages to polyploidy?
So it's quite
there's such a long
question.
There's so many answers to that question.
That's what I was going to say too.
Yeah, this is everything but basic.
Well, here's the short answer. We don't know. But I mean, it could be, the answers range from contingent selection on small part of the genome, opportunism, ecological selection based on things like elemental expense and demands for nitrogen and phosphorus. They're all over the map.
I mean, establishment of polyploidy isn't necessarily easy. You know, when you become polyploid, you know, usually the thought is it's through unreduced gamete formation and, you know, fusion of those gametes. You know, you are now the only polyploid in your population. And so if you can self, then you might have a shot of persisting long enough to make some offspring and, you know, stick around. But, you know, the first hurdle to get across is whether or not you can actually establish. Then you can do all of these cool things like adapt to, you know, niche habitats and things like that.
I get that, but it seems like polyploidy disappears with time. It's very useful in terms of creating new species, particularly in an allopatric situation, or excuse me, a sympatric situation. But then over evolutionary time, it seems to regress to a diploid state. Am I correct in that, or?
Absolutely, everything diplotizes eventually over time. It seems like, yeah.
In a lot of cases, well, Mike could also talk probably more about this than I could, but over time, yes, a lot of the genome sort of winnows its way down, but a lot of ancient polyploids maintain duplicate copies of some portions of the genes.
Mm-hmm.
That's how we get a lot of these, multiple gene copies existing. So if things are advantageous to retain, or oftentimes genes are retained if those genes interact with other genes and sort of, stoichiometric ratios. In that case, you'll keep those genes. So even things that, look today like diploids still retain oftentimes a strong signature that they used to be polyploid.
I think we could ask other way around. What would be needed for polyploids to be permanent, to like really retain the true polyploidy for long stretches of time? And if you imagine the 4 copies of the genome, you would require all 4 of them to recombine without any preference between each other. That's the only way how to maintain like a real polyploidy, right? So if over time there will be just a small buildup of differentiation of the 2 and 2 chromosomal sets. It will just necessarily have to, over the long stretches of time, re-diploidize, right? So it's sort of like a continuum, I guess.
Yeah, I would also sort of add that I think that to me, this is actually, Paul, it's a great question actually as we start our second half here for end of the 2020 discussion. Because in some ways, for me, it's the like one of my motivating questions personally for studying polyploidy. And it's also in the same sort of line of why are diploids? Like, why aren't we haploids, right? And sort of asking, looking at this question from the other direction of why do we see the distribution of ploidal levels among eukaryotes that we do observe? And I don't, as Jonathan said and other folks, sort of everybody has an opinion on this. I do think that it's a motivating question for much of the polyploidy research that we are all of us as a community investigating or all working towards understanding that question. I do contend to think and agree with Camille that for most of these situations, I think that at some level it's probably very difficult from a population genetic perspective to keep polyploids going indefinitely. That just because of the way that recombination works and getting all those alleles exposed, so that you don't just build up a bunch of null alleles, right? Eventually, just sort of neutrally over time, even. And how deterministic that process of diploidization is, I think, is still an open question. I think we have a review coming out, an annual reviews on diploidization, and we show some different evidence that there's a lot of phylogenetic variation in this, and that there's clearly room for evidence that it could be very deterministic in some lineages and less so in others. But I think teasing out those processes and what's happening with going back to a diploid state, and then why things end up hanging out in a diploid state, are really sort of fundamental questions of eukaryotic biology, not just sort of this thinking about like why is sort of polyploidy is a niche that's interesting. So yeah, and Joel's chiming in there that there's these deleterious consequences of having big genomes, and absolutely there's a whole bunch of stuff happening. And that's the other thing too with polyploidy is that fundamentally there's the genetic sort of part of this where you're changing the behavior of how selection sees those alleles. That varies between autos and allos and that whole spectrum of divergence there as well, and how recombination works. But then there's all this other stuff that happens, like changes in dosage, changes in the size of those genomes, as Jonathan mentioned too, the amount of resources that you have to use to maintain all that stuff. I don't think there's any one thing, probably, at least we haven't found that one major driver, I think, yet of why things go back to being diploids. I'm not sure we're there yet. I think it's likely to be sort of multiple things personally, but yeah, I think that's my take on it, at least. So, and Justin asks, is it primarily dictated by selection or genetic drift? And yeah, exactly. That's the question. And
that can be a whole box of worms.
I mean, we just wrote a review trying to, where we closed on that, right? We just wrote this big review on this saying that, yeah, we don't really know. I mean, how much is deterministic processes dominating that?
Right.
In the absence of selective retention of duplicated material, deletion is happening all the time. There may
Exactly.
be sort of mechanistic biases towards deletional processes anyway. So to return to a pseudo diploid state is sort of might just be a background mechanistic biophysical consequences of all that duplicated genetic material. We need selection to counteract that.
Yep.
So complicated question, Paul, with explanations from the genetic mechanism to the ecological, just have been mentioned in a few words. But it's a good one.
It's a perfect question.
Like why is polyploidy more common in plants and animals, or is it? I mean, that's the same kind of, wow, what a question.
Yeah, I mean, these are Paul. These are comp questions. These are the kinds of things I ask students during their comprehensive exams. Because I mean, these are though really it's an unanswered question, and I think understanding that question is going to be important. Is a major goal. Yeah, I mean, I'm with Jonathan. I think that I was in the office here today and was looking at my chalkboard. I had these sort of curves drawn on the board for rates of diploidization. If you have no selection, if you're strong selection, sort of what you expect those things to look like. No one's really modeled that, in my opinion. I don't that I know of really well. And there's a lot of really basic work to do there still. I think that one of the things about this, though, that's exciting about this era that we're living through is that we now have the ability to get the sorts of genomic data that for these sorts of questions, I think we really need to have from a population scale and across phylogenies to be able to get insight into these processes. Because the diploidization is the sort of a process that plays out, it plays out at different scales, and you need to be able to do that sort of comparative genomic work at different scales, but also with population data, I think, to really get at the forces that are driving some of these, to really test those hypotheses, to distinguish among those hypotheses about what's driving diploidization, and the contribution of those, the relative contributions of each of those things. So anyway, yeah.
Well, yeah, and we think of, a typical diploid is a fixed diploid, where really we have a bunch of screenshots of diploids that are all through the, continuation of the diploidization process. So even, the most traditional diploids that we think of may be more diploid 1,000 years from now or 1,000,000 years from now, even though we think of them as, very standard issue diploids today.
In that context, and back to Veronika's talk, imagine that if you had autopolyploid serpentine populations that had evolved reproductive isolation, and now we're 1,000,000 years out and we've lost any connection to the ancestors, but we've subsequently, it's been diploidized, and now you're stuck there with a diploid on a mountain somewhere in a serpentine habitat, and it has vestiges of polyploidy in it.
I was going to add, even us as a sort of example of diploid, we still have vestiges of our the last ancient tetraploidy in our ancestry. Even in human genomes, and there's still this legacy of maintaining, from I.P. McLysot's work, maintaining those paralogs to maintain the dosage balance at a small set of those genes that we still inherit from that ancient tetraploidy. Bad things happen if you change those. So there is this baggage that then comes along. So I think you never quite go all the way back to the diploid eukaryote or whatever that the haploid, I guess, but and what that looked like. So yeah, that's a great question though. So
yeah. I think we all need t-shirts that say, I am proudly proclaim, I am a paleopolyploid.
That's right.
Kind of a fractionated shirt though, like bits and pieces are duplicated, and there's big holes, gaps, gashes in the shirt elsewhere.
So this conversation made me wonder about something that maybe somebody here knows already, but I'm wondering whether our perspective would be that when we have long series polyploids, groups in which we go from not just diploid and tetraploid, but on up, whether or not those groups are different because of origination opportunities or retention opportunities. And my guess would be that there are origination opportunities, that if we contrasted the frequency of unreduced gamete production in the tetraploid level of a long series, we might find something different than what we see in the tetraploid of a short series. for example, right? That's what I mean by origination opportunities. But specifically to Mike, I guess, is this, are long series mentioned in your review about retention? And do you think, any potential to inform this question of the limits of retention?
We didn't really get into it too much in our review. We're covering a lot of ground already, trying to cover sort of the two different, as Dirk says, the two parts there of, the loss of genetic material and the loss of chromosomes as you get back down to diploidy, as you diplodize. It's a really good question. I do tend to agree that it's probably about opportunity for origination. We did some biogeographic work in the past showing that, it appears that the biggest driver of, in North America and ferns at least, of allopolyploids being present is the potential overlap of parental species. And so I tend to agree. It's very interesting to think about how this works. Like in our brassicas, where we have this ancient hexaploidy, I think some of the stuff that we're seeing with patterns of gene loss and variation, all this stuff I'm talking about is on preprints or diplodization reviews up there, and so is this paper that'll be out to a new phytologist soon. I think some of it may be because it's an ancient hexaploidy, and you've got 3 copies of genes that are diplodizing. There's this more time that we see more variation, for example, in those genes, and I think some of that might be because it's a hexaploidy rather than a tetraploidy, and it doesn't just instantly go back to being having 1 copy. And so I think there may be this more potential for these higher ploidal levels to have more profound effects over a longer period of time in that sort of post-polyploid while you're diplodizing period. But I don't know, like if we look at all the ancient polyploids that we've inferred, we don't know what the ploidal levels of those events were for the most part. We know some of them were ancient hexaploids, like in the Solanaceae and the Compositae, the eudicot hexaploidy, for example. I think there's some evidence that the seed plant one is probably a hexaploid, although that's not public yet. We don't have but we've never seen any evidence probably. I mean, I think, right, Jonathan knows the cotton one is proposed to be super like a octoploid or something. Is that right? Or a dodecaploid possibly?
I like to tell people my cotton t-shirt has 144 ancestral genomes.
There you go.
But I think what Jeanette's question makes me think about, the first thing that came to mind, Jeanette, is genera like Solanum that has every multiple of 12 up to what, 144 or more?
Yeah.
And they're still extant. What are they doing differently than in systems where you ask about, it's maybe not just opportunity for formation, but differences in retention dynamics, background genomic processes, and all the mysteries of this. I mean, how do you squeeze all that chromatin into a single nucleus? And what does that do biophysically, biochemically to gene expression? And how does that propagate up through all the omics to make something we call a phenotype? And good grief, we're so ignorant. We're just beginning to scratch the surface.
Absolutely.
I can't think of a review or a sort of a compendium of those short series and whether or not they are kind of tips of the tree, or what the evidence is in that regard. And of course, genomic work started with, you're not going to start with the dodecaploid. Things are hard enough, but I think we are in a position where we can start to think about what questions we would want to ask about those that might inform our understanding of the dynamics of, for example, retention versus origination, which is probably a bit of an overstatement of a dichotomy, but nonetheless.
This is probably kind of a naive question that sort of is talking about what you're talking about, Jeanette, but do we know if there's a correlation between these polyploid series and survey effort? In other words, have we found all of the autopolyploids?
I don't know, but it'd be pretty easy to ask whether or not groups, genera in which there are more chromosome numbers result in the detection of a greater number of ploidies, right?
Yeah, I mean, I think that should be something that's relatively easy to answer, but I do wonder if they're just, if we're just undercounting some of these series.
Maybe.
Oh, I bet we are. I bet because not everything has been counted. And
I mean, that would be my bet too, but
And so much of it is cryptic. So unless there's been a lot of population sampling, you might see it. You know, in North Carolina, you go out and look at local ash populations. You can't even tell whether you're looking at a tetraploid, pentaploid, hexaploid, heptaploid, or octoploid. They all look the same. If you have a little hand scanning electron micrograph with you, you can maybe see differences. But God, I think there's so much variation out there that we're not aware of. I love it.
Yeah, absolutely. It's a good question, Jeanette, though. It might be kind of fun to follow up on, thinking about that sort of effort and what that looks like in the landscape. I feel like maybe in one of our supplemental figures in the Wood et al. Paper, we did something with the amount of chromosome counts per genus and the number of polyploids. There was certainly a number of species in a bar chart, I remember, and the percentage of polyploids. And so there might be some information out there to sort of pull from to get at that question. It's a really good question, though. You know, in our 2016 paper too, when we looked at this distribution of across the range of, you know, autos and allos and quotes, there were genera that only had no apparent autos, but were all dominated by allos, and the opposite direction where there's almost no evidence of any interspecific hybridization producing allo polyploids, but lots of apparent autos, and it was like a perfect distribution. You know, this beautiful sort of curve that crossed right through 50-50. And I've always wanted to, what is different about the tail ends of those distributions as well, the proclivity for producing, you know, polyploids by these different origins at those extremes. And I don't know. It's a really good question. I mean, there are just so many basic things that I don't think we have a handle on. There's so much work to do.
Thank goodness.
Yes. Well, I've also wanted to always we talked about this when Brittany was in the lab. We talked about just having a project where every week we'd go to the herbarium to measure unreduced gametes off of herbarium specimens, right? And I think like that's still like even some of these basic numbers, I feel like that would be a wonderful like project to get going, you know, and just sort of move forward with sampling across all these different groups and seeing how that varies.
A great distributed project.
Yeah, absolutely. Yeah.
Yeah, it's easier on some taxa than others. On Campanula, it was super easy because those, you know, that style with the pollen bound to it because of the, you know, secondary pollen presentation, you can just basically scrape it right off with a sample without really any trouble. So others, it may be harder to do.
Yeah, I mean, when I was doing this when I was doing my taxonomic work in ferns and Adiantopsis, I'd constantly, you know, counting spores in sporangia and constantly encountering those big basketball spores that were the unreduced gametes because they didn't have the triradiate legere that you get the scars from division. So there's a lot out there.
It'd be fun to have everybody write down the one question they think is most pressing about polyploidy and look here at the intersection. I mean, every week, and see if that changes.
Oh yeah.
How it changes year over year, all of that. You know, how you keep the answers anonymous. What the overlap with the distribution of those answers is with respect to disciplines and perspectives.
Yeah, absolutely.
May I also have a question?
Yeah, this is free, open, anybody, sure, yeah.
So I feel the type of polyploidy that is not that commonly discussed among polyploidy scientists is the tissue-specific polyploidy, like human brain cells have polyploidy. Like polyploidy is kind of everywhere in practically all animals. Mm-hmm. Have you ever heard about a system where this tissue-specific polyploidy would be germline, like a polyploidy that would be specific only to germline?
Mike, do you remember at that conference if there were any talks like that? I don't remember anything specific to germline.
Unreduced gametes.
Well, okay, yeah.
That's true. I do know that you want your liver to not be diploid. That's for sure.
Yeah.
That was a bad scene.
So
So
maybe come out there's because plants don't. There's argument about this or discussion about this, but maybe plants don't sequester a germline. And so maybe any branch of any perennial has the potential to become polyploid, and then any flower that comes from that branch will be a germline polyploidy derived somatically from its ancestors.
Yeah, I guess with plants, with the not clear separation of the germline, it's much more difficult to even think about it. But the thing is, we believe that we probably see a species where they have like a completely different genomic copy that is germline specific. It's a pretty remarkable. We found it like a few weeks ago in a fungus nut in a like a species of small flies.
Cool.
So I was trying to figure out whether I couldn't find anything. So that's why I thought that I would ask if somebody heard about something, because that's a.
Yeah, I mean, I was, as you were saying that, I was thinking, the other place where there might be something weird like this are this is my lack of familiarity with these organisms, but, like in the Hymenoptera, where you get, when the haplodiploid systems, I'm not familiar enough with how those work, but I can imagine, I have this feeling that there might be something there, but it might be worth looking into, yeah.
Actually, funny enough, this species is sort of haplodiploid from the genetic perspective. So it's doing this what's called paternal genome elimination. So males always pass to the next generation only what they have inherited from their mom.
Right.
And it seems that this germline genomic copy is avoiding this mechanism. So that's it actually looks like it started by the conflict of males and females, and the males just created their own genomic copy in germline.
Very cool. That's really neat. Yeah, that's cool. That's the kind of fun stuff, like David Haig kind of things, right? So yeah.
Actually, it started by testing his theory that these germline restricted chromosomes are derived from sex chromosomes. So this was his theory, and now they're not.
So one more, I have a comment about this that I wonder what the rest of you think about. It seems to me like I'm personally guilty of ignoring endopolyploidy whenever it crosses my desk, and except that I collect the paper. So endopolyploidy is everywhere in plants. I mean, there's a recent paper in maize where they systematically surveyed all the tissues of maize for endopolyploid. And we know sometimes endopolyploid cells are responsive to stress, for example, in Arabidopsis more than some other relatively diploid cells. But I think our polyploidy community, as we've construed it here, is oriented more towards biodiversification, ecology, systematics, evolutionary biology. And so we tend to ignore it because we don't immediately grasp the connection between all this endopolyploidy stuff that comes our way in our Google searches and our fundamental interest in evolutionary biology and ecology. There's probably a lot more there, at least than I've considered, but I'm wondering if that is why we as a community tend not to have sessions on endopolyploidy at our meetings, for example.
Yeah, I tend to agree. I think that a lot of us in this community start off as from a systematics kind of background, right? And so there's this focus on the sort of evolutionarily consequential parts of polyploidy just inherently. But I do think, Brittany and I were at this meeting a couple of years ago. It was supposed to happen again this fall, but it'll happen next fall up at Bar Harbor on polyploidy. It's like polyploidy and organ repair and development.
Yeah, polyploidy development and disease, I think, was the working title.
And I think it was us and like Jeff Doyle that were the plant people from our world that were there. And I'll be speaking at the next one. So I think we're doing it next October is the plan, assuming everything's back to some semblance of normal. But most of the talks there were on endopolyploidy, and it was really fascinating to talk to. I still have email interactions with some of these folks that are, MD PhDs that are working in cancer, like this liver example where tons of endopolyploidy is really relevant, it seems, to human biology. And so I think there's a lot of interesting interaction there for crossover with some of the biology that we see with, particularly with gene expression and dosage and all of this, right? So I think there is interaction, and I think potentially on the ecological side, there's potentially way more possibility for endopolyploidy to play a role in plant ecology as well than we sort of probably give it credit. I know that like the example out here that people often use locally is the saguaros, the parenchyma, or like octoploid or something, that are associated with water storage. And there's some evidence this might be some nucleotypic effect, but anyway.
That's, I think, what is coming up is a technological way, because we need single cell transcriptomics, and that is not used in plants yet.
That's right.
Rob Laporte put an interesting comment. I think he's suggesting a group review. You put a title in the chat. Rob, you want to explain, or do you just leave it like that?
No, I think you're about right. I was just kind of thinking like a symposium or a review or something. I mean, I think that the endopolyploidy is typically overlooked. I mean, I can't say that I have focused on it in any meaningful way, but I'm starting to wonder about this sort of apparent lack of differentiation of the germline or the sequestration of the germline in plants, and how endopolyploidy may be leading to a lot of this, macro mutational event that we see.
Yeah, there's certainly examples of that. There's like an old paper from 1948 in AJB, Butters and Tryon, I think Tryon was the other author, and Woodsia in a fern where one frond, during frost had damage and it became a tetraploid, and then, gives rise to all the spores are unreduced, right, produced by that frond. So there's certainly that kind of potential, but think about establishing even polyploid plants. But yeah, I think it's cool to think about the ecological consequences of endopolyploid changes. That's a whole black box, I think, right, that we haven't really explored.
Not to needlessly complicate that black box, but I was just thinking about, all of the chromosome counts that we have and flow cytometric estimates for ploidy for various things. And, some subset of them come for things like pollen mother cells and things like that. And then others come from leaf tissue. And the ones with leaf tissue, how many, what subset of those are actually just, endoreduplication along that branch, rather than the entire individual that we're just assuming is polyploid.
But with flow cytometry, you would see that. That's what we frequently do with classes to do flow cytometry on tomato seeds compared to kiwi seeds, where you can nicely see in the tomato seeds the multiple peaks. More worried about old chromosome counts.
Such an interesting discussion.
Absolutely. Yeah.
I had a thought. We know that endopolyploidism, if that's the right term, occurs under situations of stress, whether we're talking animals or plants. Is it possible that we're observing natural selection on a cellular basis, where endopolyploidism is occurring naturally, spontaneously all the time, but that it doesn't become evident or is not allowed to express itself except under these stressful situations? In other words, for instance, that fern that suffered frost damage, maybe there was endopolyploidism on a regular basis, but it wasn't an advantage until the stressful situation arose.
I think that's some of the thinking with the herbivory hypotheses, that just the ability to add tissue on rapidly may be one of the sort of lines of cell line selection that favors endoreduplication in those situations. But it's also likely to change all those things we know relate to cell size versus nucleus size, right? So, yeah. What the advantages might be, how they play out, at a physiological level, I think is mostly black box.
Yeah, there was a guy at the Polyploidy conference last year, Don Fox at Duke University, who did some work on wound repair systems in Drosophila, I believe. And he showed that polyploidy was necessary for wound repair. And there was a whole new mechanism of chromosome division, I think, that allowed tetraploid chromosomes to divide. That wasn't the standard, metaphase and division that actually prevented quadrivalent formation. And so he's done a lot of work to see, even though polyploidy is so detrimental, there are ways around it if you need to. Which also brings up the question, if in animals that have sex chromosomes, if they're so resistant to polyploidy in the germline, how can they tolerate polyploidy somatically? How can they tolerate endoreduplication if they can't tolerate it in the whole organism? Which I think is another huge black box that we don't.
Yeah, absolutely. Absolutely. That's one of the classic arguments, right? Again, about why polyploidy is rarer than animals. An old one is that their developmental system is too complicated to handle the polyploidy. I'm like, but endopolyploidy I think really sort of kills that one. It makes it much more about probably genetics and sex chromosomes and everything else. But Don Fox, by the way, is the person that organized this meeting in Maine, and so. For those of you that are interested in this line of questions and having it's like a parallel universe. It's like going to another dimension is what I felt like when I went to that meeting. Maybe Brittany can express that, talking about polyploidy to medical doctors. And it was like going into another world. And, all of us trying to figure out if we're using the words the same way.
Might try polyploidy in cancer.
Yeah, exactly. Yeah.
Just in those papers you were talking about of Don Fox's with the way the sort of chromosome can sort of know who they sort of originated from and keep track of their division that way. When I heard that at this meeting, I immediately started thinking about autopolyploids in plants. And, we know that autopolyploids exist and are common, and for a long time the thought was they weren't as common because multisymbolic inheritance would mess everything up. Maybe plants are doing something similar. I think it'd be a fascinating thing to look at.
Well, right, and that brings us back to the diploidization process too from autopolyploids. How do chromosome pairs recognize each other? Because at some point you have to stop pairing in quadrivalence. And is that just because there's enough differences between, two different sets of homologs so that they get proper pairing? Or is there some learned mechanism of dealing with that separately?
Yep.
I'm wondering if we're going to have jobs for a while.
Yeah, there's some genetics. We just reviewed a bunch of that stuff in this review. And there's some genetics that appear to be somewhat common, although I know from other things that aren't quite published yet from other folks that it's not, that clearly they're not also common everywhere else. How's that? But I think that it also evolves pretty quickly. It seems like in a lot of these cases that you get meiotic stability pretty fast, and otherwise you just don't make it, right? And but yeah, it's a super relevant question as to how that process works and how much this is adaptation versus changes in sort of adaptation in the meiotic genes themselves and the pairing mechanisms versus ultimately sort of eventual structural changes that lead to distinct homologs that no longer pair quite so easily, without having more negative consequences for the organism. So it's all open questions. We got in trouble for our in this review on diploidization for saying that we don't know. We got in trouble for having the phrase "we don't know" too many times, so we had to come up with different ways of saying that, basically. But I think that's so true that we know very little about some of these aspects, even in the stuff that's really recent, like in neo tetraploids and things like this. We just don't know. But more fundamentally, we don't have a great understanding of how homologs recognize each other in general. I mean, it's basic biology. And then we're layering on this really this other complicated layer of biology on top of that, and asking questions through that lens. And I think, there's a lot of fun stuff to do interacting with folks that are just basic molecular biologists on pairing behavior. That there's some really fascinating things as a polyploid biologist, I think, to do there, in a whole sort of multiple careers worth of research on those axes. So yeah, certainly we're not out of jobs anytime soon. So that's been said a few times. So
I have a question that kind of, I think, loops back to something Paul may have mentioned. So talking about endopolyploidy in response to stress, and just polyploidy in response to stress. And, there was the Rice paper in 2019 that looked at sort of, the broad sort of geographic pattern of polyploidy. And there's sort of long-term been this idea that there's more polyploidy in cold climates. So what do we think is going to happen to polyploids as climate change progresses? Is polyploidy induced or encouraged by stress, but any kind of stress, such that we would expect that in hotter, more stressful environments, we would see more polyploidy? Or is there something special about cold climates such that we would see a decrease in polyploidy? If that makes any sense.
No, it does. I just don't think there's a simple answer to it. I mean, there's a huge number of polyploidy events that correlate with the last mass extinction event.
Mm-hmm.
So it looks like polyploids may be better off than diploids, but whether I don't know if there's a environmental correlation with that or not.
Just seeing if anybody had wanted to go ahead and place their climatological bets now. So.
I think so. There's a paper at least on unreduced gamete formation in natural plant populations. The paper by Julia Kreiner and out of
that's a great one.
Yeah, I've read that one.
Ryan Husband's lab, and the thing that struck me there is that we have no real data on the plasticity of unreduced gamete formation and therefore its association with climate. And so if you want to think about how things will change under climate change, well, of course there's adaptation, but the first line of action is going to be what's the plastic response to different environmental conditions. And we have incidental reports and accidental experiments, the Lilium stuff from whenever that was, with the greenhouse being getting cold by accident. But we have very few actual experimental studies that would show variation in unreduced gamete formation under different environmental conditions.
So this goes back to what Brittany and I originally talked about, I think, with the herbarium scan, was to look at not only unreduced gametes across different species, but also across time. In different populations. And you might be able to sort of do how sort of like folks like Chuck Davis and other folks have looked at shifts in phenology, right? You might be able to detect changes in the last couple hundred years.
Yeah, let's do that.
I'm serious. No, I mean, really, I'm all about this. So partly this was a way I was trying to think of how I can get myself back at a herbarium more instead of sitting at my computer. And this would, I feel like it's a really basic fundamental question. So yeah, it'd be fun to do.
Oh, that's a really interesting thing you just put in the chat. I had no idea that happened. That's cool.
There is a really similar phenomena with asexuality that they call it geographical parthenogenesis, that on the more northern regions and more extreme environments, there is much higher abundance of asexual species. And they have a bunch of explanations for that. Some of them are guessing that it's just easier for asexuals to colonize north, and somehow it seems that for polyploids too. So perhaps it could be just simply the population dynamics that is better for asexuals just because they don't have to search for mate, and in tetraploid or like polyploids because they just grow faster if they have more expression. But there are other hypotheses that are saying that it's other way around, that there is too much competition on South, and that's why asexuals get faster outcompeted. I wonder if like if it does have a complete parallel to polyploidy or not, but it sounds like a remarkably similar problem.
Wouldn't it be simply the lack of pollinators in colder climes would disadvantage sexual reproduction?
Well, these are mostly animals. So usually when we talk, when I talk about asexuality, I think about obligate asexuality, which is actually really rare in plants. Like those that are doing the asexuality in plants usually still do sperms and fertilize the sexual relatives and create new asexual lineages. But this kind of like a real parthenogenesis of genomes that are never ever recombining is just like an animal thing.
All right. Well, this has been wonderful, but I actually have another meeting coming up pretty soon in my day. Well, I think this has been great. We should maybe, as I put together the spring schedule, build in a couple sort of open discussion times like this, and make sure that Paul Allen arrives to ask us those basic. Though those are the most basic questions that really motivate this work, and it's just great.
Well, thank you for having me.
No, thank you for joining us today.
Can I put a shout out on behalf of the whole community to Mike for starting this series? And I think we all ought to tweet out our gratitude and our how much we look forward to a continuation of this series. It's been over the years, over the last 20 years or so, we've been having these international polyploidy conferences, and it's always a reminder, these conferences are always a reminder of what a terrific community we have. Open, supportive, smart, interesting. And I'm looking at Camille here and I see a bottle of Ardbeg in the background and Redbreast. And
yeah, I was seeing that too.
I'm just looking forward to what you can share. But anyway, thank you, Mike, on behalf of everybody, including the hundreds of people who participated and who are not here today. But let's tweet this out. Who's going to get it started? And then, then we can all retweet. Can one of you get it started?
Well, thank you. Anyway, thank you, everyone. This has been fun to do, and it's been, I think, for me, it was just a way to try to stay in touch with people outside of my house, and so it's and to hear about great research from all the meetings that we would miss, and I have a couple of new students that I wanted them to be able to get a good dose of this kind of stuff, and so it's been really, really fun for me. Hopefully, everyone's happy with the format. I mean, this is just sort of what we're doing. It's been easy to organize in the sense that all I've had to do is post these requests, and I've had to poke people a few times to get people to talk. So let's try to volunteer to speak. How's that? And, if you're students and things, and hopefully we will be able to keep it going without a whole lot of work. The community is easy and wonderful to work with, and as Jonathan said, I look forward to hoping to keep these moving forward for the rest of the year. So,
Mike, I really applaud the focus on early career graduate student, postdoc, early career scientists as well. It's a wonderful venue. They're
doing the interesting stuff, and I think especially now we want to be able to highlight the work of those folks. In this environment, the meetings are so important. Networking is so important, right, for career development, that it's important that they get a chance to show what they're doing to the rest of the world. And I know those videos are watched, 50, 100, 200 times. So, these, not just the folks that can join us live, but also those YouTube videos that are now posted of these talks. Hopefully give people a chance to go back and revisit this material, and folks that didn't get the chance to join can also see it. So I think that's really important for people's development. Yeah. All right. Well, thank you, everyone. Yeah.
Mike, thanks a lot. This has been great for keeping people together, and it's been really informative. I think, as I think Jeanette said it earlier in the comments, it's been one of the bright spots of 2020. So thanks a lot.
Oh yeah, you're welcome, everybody. For the same here. I mean, it's been awesome, and everyone in this community is so great to work with. That it's just fun. Thank you.
All right. Bye-bye, everybody.
Bye.
Bye, everyone. And as
Bye,
usual,
thank you.
as usual, I'll post this recording up here on the website and Twitter later on here once it's available. All right, everybody. All right. Have a good day.